Of a protein called actin. Let us see the structure of thin filament, or actin filament in detail. Thin filament, or actin filaments are made up of 3 different proteins. 1. It contains 2 polymeric filamentous, or F, actin filaments, which are wound together. They are a polymer of globular, or G-actin monomers. 2. Tropomyosin filaments are coiled around the actin filaments lengthwise. 3. Troponin, which is present at certain points on the tropomyosin, covers the active binding sites for myosin. Troponin and tropomyosin regulate the binding of actin and myosin filaments, hence play an important role in muscle contraction. This diagram shows the structure of actin filament. It shows 2F actin made of many globular proteins, shown as light green colored spherical structure. The orange-colored thread-like structure is the tropomyosin filament. It run parallel to F filament. Troponin is represented as reed beads of group of 3 intermittently. Now, let us see the structure of myosin filament or thick filament. Thick filament, or myosin filaments are made up of the protein, myosin. It is a polymeric protein, consisting of monomeric units of a protein, known as meromyosin, mm. It has 3 parts, a tail, a short arm, or neck, and a globular head. The head and cross arm protrude outwards from the filament at regular intervals, forms the heavy meromyosin or HMM. The globular head contains binding sites for ATP and actin. The head acts as an ATPase enzyme. The tail forms light meromyosin or LMM.
This the diagrammatic representation of myosin filament. The head shows ATP site, actin binding site and short arm, together form the heavy meromyosin. The tail forms the light meromyosin.
Sarcomere, the contractile unit of muscle. A myofibril contains hundreds of sarcomere, which are connected end to end. A sarcomere is the basic unit of muscle contraction. It contains repeating units of actin and myosin filaments, in an orderly fashion. Sarcomeres are joined, at the end by interweaving filaments forming the Z-line. It is the limiting membrane of a sarcomere. Actin filaments, or thin filaments are attached to the Z-line, at frequent intervals. Thick filaments, or myosin filaments are present, between the thin filaments. They are held together, at their place by a very thin fibrous membrane, known as M-line. The actin and myosin filaments overlap each other, in a specific pattern, and result in the striation pattern of muscles. They form 3 types of bands. 1. I-band, or isotropic band. This is the light band, and comprises actin filaments attached to the 2 adjacent sarcomeres. Here, thick and thin filaments do not overlap. 2. A-band, or anisotropic band. This is the dark band, it contains overlapping actin and myosin filaments. 3. H-zone is the middle portion of the thick filaments, where thin filaments do not overlap the thick filaments. This diagram shows the arrangement of actin and myosin filament. The actin filament, on either side of the myosin filament, which partially overlap the free end of the myosin filament. This arrangement leave a clear zone in the center of thick filament called H-zone.